Good morning, good afternoon, good evening, everybody. We are super excited to have so many people having signed up for this integrative analysis of UK Biobank Proteomics data. As you all, I'm sure, know, the first tranche of proteomics data is out on the UK Biobank, and we are very excited to talk about it and how it got there. So, as you all know, we have a star-studded cast of speakers who are all going to present effectively lightning talks during this presentation. So we're going to start in just about a minute. I'm going to turn it over to our MC, Brenton, who's going to go ahead and walk you through all of the logistical considerations and all that. But first, I'd really like to say a big thank you to all of the presenters who took time out to be here, as well as almost a dozen folks who are on this webinar from the various institutions listed here to answer questions. So please, please, please type as many questions as you want into the Q&A. We will get there. And yes, the slides, as well as the recording, will be available after the meeting on community.dnanexus.com. With that, we have a lot of material to cover today. So I'm very excited to turn the floor over to Brenton Pyle, who will introduce all the speakers and go through all of the other logistical things. Thanks, Brenton.
Fantastic. Thank you, Ben. I want to introduce, like I said, our star-studded panel. We'll be starting with Naomi Allen from UK Biobank, giving an introduction to UK Biobank and the consortium projects, and one that resulted in the data we talked about today. Cindy Lawley giving a background on Olink Proteomics, which helped part of that consortium to create this new data set. Chris Whelan giving an introduction on the Pharma Proteomics project, and Ben Sun following up on more detail on generating that data. Then we'll be passing it over to Karsten Suhre, who will be talking about analyzing this data, and then Ben will be bringing it all together, talking about how we can analyze this on the UK Biobank Research Analysis Platform, aka UK Biobank RAP. Couple upcoming webinars that we have. One to shout out is the Oncology Researcher Roundtable, where we'll be getting us another panel of experts to discuss oncology research using large-scale data sets like UK Biobank. Then we'll be having another UK Biobank Proteomics data webinar on June 1st, and that'll be a good companion piece to that. We'll be diving a bit deeper into the RAP platform functionalities for analyzing this data. You can find all of this content in the related content sections or on our community forum. If you are not already a community forum member, this is a place for UKB RAP researchers to gather, find updates for events like this, new data releases for data that we'll be mentioning today, and also collaborate with other researchers to share pipelines, workflows, Jupyter notebooks, ways that you can make your research and pipeline assembly even easier for working on RAP. And it's also monitored by UK Biobank and DNAnexus team members to answer your questions. That is also found in the related content section if you'd like to sign up or follow that QR code. If you are an early career researcher or a researcher from a low or low middle income country, you can get platform credits to work directly on UK Biobank RAP. So you can apply via that QR code there. It's also found in the related content section. I'll be mentioning that a couple more times. And you'll be able to get credits to work directly on the platform. And also you are qualified for reduced access fees if you are a researcher from low or low middle income country and an early career researcher. If you're a commercial organization that is already working on RAP, the DNAnexus Advantage team offers their services to help create pipelines, generally just make your work easier and get you up and running on the platform, or to enhance every work that you're already doing there. So if you'd like to find more information about the accelerator package, you can scan that QR code and we'll get in touch with you for more details. If you are a community researcher and you would like to promote your research in events like this or upcoming roundtables in the newsletter Spotlight, you can submit your application now. This is recently opened, and so we love to engage our community and would love to have you speak about the work you're doing on RAP or with the UK Biobank dataset. And that application is at that QR code. And again, in the related content section. And finally, if you are brand new, you haven't heard of the research analysis platform before, or even if you have and you're approved, you may not know that it's available for all approved UK Biobank researchers. You can join as part of your approved application and receive £40 credit just for signing up. So you can register there via the bit.ly link and in the related content. And it's a way, we'll be speaking more about it today if you are unfamiliar, but it's a way to access, it's an all-in-one platform to access the data set, use a cloud infrastructure to analyze it, and also a full library of tools and the ability to import your own custom tools as well. That is it for my big introduction. I would like to pass it over to Naomi Allen to kick off her presentation.
Many thanks, and yeah, very excited to be here to talk about the proteomics data. So many people who I talk to about UK Biobank often think of it as a rather fancy biorepository for a load of samples, but actually UK Biobank is a lot more than that. It's an epidemiological study with a unique combination of both being of very large size, having a huge breadth and depth of data that we've collected on all of our participants, with a long duration of follow-up, with now over 15 years of follow-up of all of our participants through their health records to find out what happens to their health. And perhaps most importantly of all, the data is very easy to access, with over 30,000 researchers worldwide now using it for health-related research. So UK Biobank is really renowned around the world now for the sheer breadth and depth of the data that we're collecting on our participants. So in addition to the lifestyle information we collected at recruitment for all participants, we have information on biomarkers from the biological samples we've collected, MRI scans from participants coming to bespoke centres, the genomic data on all participants, which I'll talk a little bit about in a moment, a whole range of physical measures for everyone, and a follow-up of health outcomes through linkage to health records. And it's the combination of all these different types of data in one place on a very large number of individuals that is really making UK Biobank such a unique resource to do health-related research. So I just wanted to spend a couple of minutes talking about the power of open science using a resource like UK Biobank and how that's led to these public-private collaborations, of which the Proteomics consortium is just one. So first and foremost, it's the easy accessibility to the UK Biobank data to the global research community that has really spearheaded these novel types of collaborations. Because once you get your hands on the data, research groups around the world quickly realized that there are actually opportunities to enhance the resource further. So as well as the lifestyle information, the biological samples, and the health outcome data that we've got, there was enormous scientific added value for researchers to say, wouldn't it be great if we could add genomic sequencing data to the resource and other omic data sets at scale for all 500,000 participants? Because that would massively accelerate drug discovery and development work, and also facilitate research into risk prediction and stratification, and so on. But of course, cohort-wide assays and other cohort-wide enhancements require very deep pockets. And so there's a need for funding consortium to come together to make these investments to perform such enhancements. And that's led to the establishment of new models of pre-competitive collaboration, whereby research groups who invest in the resource to perform these enhancements get a short period of exclusive access to the enhanced data before they're then made available to the broader research community. So the first example of one such pre-competitive consortium was around the whole exome sequencing project, which was led by Regeneron together with a consortium of other pharma companies, where they performed whole exome sequencing for the full cohort. So the data for the first 50,000 participants was made available in 2019, with tranches thereafter, and data for the full cohort was made available the middle of last year. The success of that consortium then quickly led on to a public-private partnership together with charity, industry, and UK government funding to perform whole genome sequencing, again, on all 500,000 participants. Data for the first 200,000 was made available the end of 2021, and data for the full cohort will be made available the end of this year. So UK Biobank, we've been focused over the last couple of years on really maximizing these opportunities of turning these depletable samples that we have into reusable data. The Genomics is the first example of that. But there are now other assays underway, such as transcriptomics, where we started a pilot to do single-cell RNA sequencing on 5,000 individuals, proteomics using the Olink platform on a pilot of 57,000 participants, which is the subject of this particular webinar and is of huge interest to the research community. And also metabolomics, performed by Nightingale Health, which is performing NOR metabolomics on the full cohort with data for part of the cohort already available. And so you can see there's increasing momentum now to do these type of enhancements being performed by a wide variety of external funders. And so you can see that over the future, there may well be potential to scale these up to cohort-wide assays, to perform different types of assays, so targeted as well as untargeted approaches to metabolomics, proteomics, etc. And also using other types of assays such as epigenetics, immune system biomarkers, and so on. So there's a huge interest being generated in this area as to what cohort-wide samples can be used for these types of assay generation. So in addition to focusing on the sample assays, we've also been focused over the last few years in setting up a project to bring participants back to dedicated imaging assessments. So we want to image up to 100,000 participants by the end of next year. We've already done 70,000 participants where they undertake a range of MRI scans. And we've just started a repeat imaging study where we intend to bring 60,000 of those 100,000 participants back for a second scan, which will include full repeat of the baseline assessment, plus some additional blood samples and additional eye measures. And of course, the beauty of having repeat imaging measures is that it will enable researchers to assess longitudinal changes in the structure and function of organs before overt disease develops. So we're hugely excited about this. So as I mentioned before, UK Biobank is a longitudinal study, so we follow up all of our participants via linkage to key electronic healthcare records, and we have done for the last 15 years. And you can see here, by the end of 2020, which is the last date of complete follow-up, you can see that we've got thousands of incident cases of the most common diseases. And over the next 5 to 10 years, there'll be substantial numbers of the more common and even less common diseases to enable really robust research to be performed. So I just want to finish by highlighting the 4 unique combinations that really make UK Biobank one of the world's best health-related resources. And that's the scale, the depth of the data that we've collected, the duration of follow-up that we have, and the ease of accessibility of the data. And I just want to end by acknowledging the staff at the UK Biobank, all of our scientific advisors, our core funders, and perhaps most importantly of all of our 500,000 participants, without which this simply would not be possible. Thank you.
Fantastic. Thank you so much, Naomi. And now to our next presenter, Cindy Lawley, who will be giving an introduction to Olink.
I'm here to talk just a little bit about Olink, just a few slides in here. Love listening to Naomi's context of what these proteomic data are included in a massive data set and a revolutionary set of samples. I'll talk a little bit about the foundation of our technology and the data type that we're working with. The foundation of Olink's technology is illustrated in this video loop. This is what we call the proximity extension assay, or PEA assay for short. we're essentially finding proteins in solution using 2 antibodies per protein. The protein in this video is illustrated as a blue rough circle. Once both antibodies find the protein target, they're able to then hybridize 2 oligos, single-stranded oligos, 1 per antibody. And once that hybridization happens, it's sort of a 3-factor authentication that can be extended, then amplified, and be a reflection of the original protein level in the sample itself. So effectively, we're converting a protein signal to a DNA signal. And that DNA signal can then be read out on a qPCR instrument, or in the case of the data that are represented in the research analysis platform, data coming out of the NGS instrument. So today we're able to do this for around 3,000 proteins. We have plans to, of course, expand that library and cover more of the proteome, and we've done this in a modular fashion. But I want to explain kind of how our data complement the data that are traditionally thought of as the gold standard. Mass spec methods are really considered the gold standard in proteomics. So what does Olink add that's unique in the world of proteomics? So I like this image because it sorts proteins in this. This is our first iteration of our Explore library. That's our NGS readout, the product line with the broad coverage of the proteome. And here we've got 1 dot per protein from the highest abundance in the upper left to the lowest abundance in the lower right. The units are along the right-hand side from above milligram per mil to below picogram per mil. So the first iteration of our library has this emphasis on this, as where I've put the box, on this low abundant density. You can see the density of the dots, the thickness of this. And of course, for each one of these dots, you're going to have some dynamic range that you're going to be able to detect these samples in, healthy versus diseased samples, so that you can imagine little whiskers on each of these dots. But just for simplicity, focusing on this low abundant plasma proteome has really been enabling to help allow us to have a look at an area where it's just not simple to do large numbers of samples using mass spec. So the view in this low abundant plasma proteome, I think, has really enabled the number of publications. We've seen over 1,200 peer-reviewed publications just since 2016 on the Olink platform. So what's the data look like? What's the output? The output file, if you were to do a project with us, looks somewhat like this. It's called an NPX data file. It stands for normalized expression data. Our assay is a relative assay. Those flat files are pretty easy to handle. They're easy to manipulate. The data, I think of BAM files and sequencing files. This is a pretty straightforward format, and they're delivered in a log2 scale. So what that means is a single NPX difference in protein level is going to equate to a doubling of the relative protein concentration. So a great feature, of course, of the log2 scale is the normal distribution, roughly normal distribution. So it's quite compatible with standard statistical tools, t-tests, ANOVAs. And of course, our internal team of scientists is always keen to see how to accelerate people's ability to make sense of these data and make discoveries. So my last slide is this one, where I'll talk about some of the tools that Olink has built that are freely available for folks to look at the different aspects of their data. So the first is Olink's statistical analysis app. This is a web-based application. It includes an ability to drag and drop data files, run t-tests, ANOVAs, generate figures like this volcano plot, and you'll often see figures in publications that come out of this app. The second is the R package. Now this R package is on CRAN and includes similar or same tools that underpin the web app, plus additional functionality. It allows time series analyses, some examples shown here. And I hope Ben will talk a little bit about how an R package like this can be integrated into DNAnexus or any existing pipelines. And finally, the tool that I use most is something called Olink Insight. This is one of our newest products that we've offered as a web-based application. This incorporates many of the available open access sources in one location. It includes the Reactome database that can be navigated. It includes resources from Uniprot, from Go, or Gene Ontology database. So we have also unique data sets in there just from Olink. This allows for you to explore the natural variability of the proteome. We have some healthy sample data in there as a reference. What I like about it is you can put in a gene list. Often I put in gene lists that are relevant for polygenic risk scores and convert that to protein lists and review publications and Go terms and share it with customers. So I encourage anyone to check it out at insight.olink.com. And then, of course, if anyone has any questions about the work that has been done here or anything about the data types that Olink offers, please feel free to reach out to support@olink.com. Thank you so much.
Okay, terrific. It's lovely to be here. I'm going to provide some additional context for why we formed the Pharma Proteomics project, why 13 pharmaceutical companies came together and agreed that the generation of this unprecedentedly large data set was a good investment. And so I will start by probably stating the obvious that UK Biobank is one of the largest and most detailed health studies ever undertaken. Its depth and breadth is just absolutely breathtaking. But industry scientists like myself and like Ben Sun, who you'll hear after me, are using population studies like the UK Biobank to drive a process that we broadly call genetics-guided drug discovery, or G2D2 for short. So with G2D2, we systematically study gene variants across very large populations to find associations between genes and diseases. And once we validate these associations, we can take them to our biologists and to our chemists within pharma and try to turn them into new drugs. And there are very few ways to predict whether a drug program will be successful, but genetics happens to be one of the very few strong predictors. There have been several peer-reviewed retrospective analyses of drug development pipelines at companies like AbbVie and AstraZeneca, and those studies have shown that for drug programs that make it to the patient versus those that don't, the ones that were successful were at least twice as likely to succeed. The ones that were successful were at least twice as likely to be supported with evidence from human genetics, be it GWAS or OMIM or other evidence. So there's evidence to suggest that this is a strong process. However, genetics is not perfect. It usually points towards gene regions, not exactly the exact gene. It often points towards correlations, not causation. We often have to do downstream Mendelian randomization to figure out the causal influence. And sometimes even when we can identify the causal impact, the actual biological impact is not entirely clear. So this is where proteomics can come in, it can really help. If we add multiplex proteomics to this G2D2 process, we're actually integrating the proteins, our actual drug targets, into the process, and it could enhance this process overall. And so just to even go back even further, what do we mean by proteins? They're the building blocks of life and they're the building blocks of drug development. We not only use them for this genetics-guided drug discovery process, we also use them to, in many cases, form our biomarkers for clinical trial programs. One example at Janssen is plasma p-tau217 for Alzheimer's disease. We're actually using this in some of our tau trials for Alzheimer's. But there are other everyday examples like ApoB for familial hypercholesterolemia. And folks like Karsten, who you'll hear from later, and Jochen, Suhre, Jochen Schwenk, have all agreed that proteomics is starting now to take its place alongside genomics for the comprehensive characterization of human health and disease. When we say multiplex proteomics, broadly speaking, we can talk about targeted proteomics, which typically takes a patient sample, runs it through an immunoassay, which is developed to detect a single protein and typically detect it at very, very high sensitivity and specificity. And then with multiplex proteomics, again, very broadly speaking, you can have mass spec-based multiplex, which can capture many different proteins at the same time by measuring the mass to charge ratio of ions. in a sample. However, the main limitation of mass spec, despite being seen as the gold standard of multiplex techniques, is that it often requires very large patient sample volumes. It often misses lower expressing proteins like inflammatory cytokines, and it's not particularly high throughput, which is where the affinity-based techniques come in. The antibody-based technique like the one developed by Olink, or aptamer-based techniques like those developed by SomaLogic. These are still quite specific and quite sensitive, but they require much less patient fluid, and they can detect lower expressing proteins like those inflammatory cytokines. And recognizing that there was a lot of potential for these multiplex affinity-based techniques, we formed a consortium called the Pharma Proteomics Project to fund the generation of Olink-based, antibody-based proteomic measurements across over 57,000 individuals in the UK Biobank. So this is generating just under 3,000 measurements, or the first 1,500 proteins now publicly available. And the reason that we formed this was to overall identify better genetically validated drug targets, find better biomarkers, and make better medicines. So this was a 1.5-year project. We announced it in November of 2020. The first phase commenced in April of 2021, and that's when we decided to conduct the first batch of 1,500 proteins that are being released this month. However, about halfway through the project, Olink doubled the number of proteins that it could measure. So we initiated a second phase of the study in November of 2021, and that extended the project to the Explore 3072 platform. And we now have data on just under 3,000 unique proteins. That first phase for the first 1,500 completed in December of 2021. And we submitted a preprint white paper to bioRxiv in June of last year describing these first 1,500 proteins. Phase 2 for those next 1,500 completed just at the end of 2022. That brings us to just under 3,000 proteins in total. The first phase release, obviously the reason we're all here today, is happening this month. The second phase release, I believe now, will be sometime in November. And this does obviously represent the world's largest open access proteogenomics data set. So how does this break down overall? This is about 10% of the full UK Biobank cohort, and we wanted to make sure that this was broadly representative of UKB as a whole. So most of the participants in this data set of currently just over 54,000 individuals is randomly selected. It's just about 46,500 people who were completely randomly selected. However, there are some cherry-picked or pre-selected individuals. The first set come from the COVID-19 imaging repeat study from UK Biobank, just over 1,200 people. The second batch then is a batch of pre-selected or cherry-picked participants from each of the pharmas. So all 13 companies pre-selected 500 individuals. So for example, if Biogen was interested in a particular neurological indication, they could pick that. If Pfizer was interested in a particular immune indication, then they could pick that. And as you can see on the graphs on the right, the randomized cohort is broadly representative of the full UKB cohort. And as you might expect, the pre-selected cohort is enriched for certain diseases of interest to the pharmas. So what have we done with these data so far? This is my last slide. We've done quite a bit. I think, as I mentioned already, we've published a preprint on bioRxiv. It's called Genetic Regulation of the Human Plasma Proteome, and that describes the GWAS of the first 1,500 proteins. We've recently updated that to include all 3,000 proteins, and fingers crossed that will come online in a peer-reviewed journal sometime over the next few months. But beyond that, a lot of other studies have come out in the public. The AstraZeneca and Biogen teams, for example, leveraged the exome sequencing data from UKB to study rare variant high impact associations with these proteins. And again, that's on bioRxiv for those that are interested. Another bioRxiv publication that recently came online was a study of neurofilament in ALS, or motor neuron disease, that was led by the Qatar team. And also the Calico team more recently looked into proteomic predictors of mortality. And that's just a very brief cross-section of what we're doing overall at the American Society of Human Genetics, in October of last year, there were 19 different abstracts, primarily based on this data set. So this is just a point towards how there's lots of future papers coming online, and that's just from the pharma partners. We're really excited now that this has become available to the broader scientific community to see what the community gets to do with these data. And with that, I'm going to pass to Ben, who can go into more detail about the consortium preprint that you see at the top right of the screen.
Thank you, everyone. So thanks for Naomi, Sandy, and Chris for laying down the foundations for all about the UK Biobank and the Olink, as well as the project as a whole. So today I'm going to, in addition to talking a little bit about the preprint, also just talk about some of the esoteric technical issues related with the proteomics data and things to look out for when the wider research community is working with these data. So as Chris has mentioned, there is actually a nested component within the UK-PPP project. And so there's a randomized baseline component where it is a random subset of the UK Biobank that's been sampled to be the most representative of UKB. And obviously bear in mind that the UKB is not necessarily representative sample of the UK population, but it seems to do a reasonably good job, at least for the adult population and age ranges of representing the UK. And the consortium samples can be quite different to the underlying random baseline samples. You can see from the zoomed in figures that the randomized baseline does a pretty good job of approximating UKB on all fronts. And in the preprint, as well as in hopefully in a few months' time, when the data, the summary data, as also some of the details become online publicly as well in a peer review journal, there'll be additional information on the differences and similarities and representativeness between the baseline samples and the consortium select samples, as well as the COVID-19 imaging samples compared to the UK Biobank as a whole. So as you can see that the consortium select samples in general tend to be slightly older. And if you include all the UKB samples in addition to the baseline, you actually get enrichment for all sorts of diseases across the board. And obviously some enrichments seems to be higher than others as a mixture of the prevalence of the diseases, but also a function of some pre-selections that were happening. But I think this nested structure gives the scientific community a boosted power to look at the association with rarer diseases, which otherwise would not be possible if you use randomly sampling. But also it allows a component where if people are interested in data that are representative of UKB, that is also available as well. So depending on what sort of research question and the hypothesis that people are looking to answer, there's different ways and different components of the data that can be used. So that's one thing to bear in mind. And the other thing is obviously the potential sources of technical biological variation that also exists. So when we computed the CVs, actually, you can see that there is actually an increase in CV with low abundances. So from left to right is the color code essentially goes from low abundance to high abundance proteins. And you can see that the general trend for the coefficient variations is in accordance with what you would expect with the abundances, along with the LOD as well. But in general, by and large, the CVs are pretty good. Mostly are sitting less than 10% for the majority of the proteins. So the technical reproducibility and repeatability of the assays seems to be reasonably in line with what's been previously reported. And of course, the below COD threshold, the below COD values are actually also reported. So obviously care should be taken when analyzing those proteins. But obviously, that doesn't mean everything below LOD, the values are just complete noise. So there might be some informative elements within those below LOD values as well. And comparisons, when we compared some of the biological associations, we see sensible proteins as well, as people might see in the preprint. But when you compare the association with previously reported studies, bear in mind that different covariates were used to adjust for things, and different normalization and QC procedures were made. You can see that for the red ones, where we're comparing Olink with Olink, you get quite a good alignment between the effect sizes between different studies that were previously published. You also, in general, get a good positive correlation with the SomaScan assays, one of the other broader assays there, but you see the correlations slightly less strong compared to the Olink ones, which is expected given that within technology, reproducibility across cohorts and across studies seem to do better than across different technologies as well. So obviously that's something also to bear in mind when people are using the Olink data in UKB as either as replication or as comparison studies as well. And also other sources of variation to be aware of are some of the key ones we've been investigating as of listed here, with some technical sort of plots for people to see that we don't see any major clustering or major outliers that are seen between some of the age and some of the ancestries, but also the variability being assigned to some other demographic and technical and biological factors. As you can see that on average, blood count, as you might expect, probably play a slightly more role in variation of proteins compared to some of the other demographic and seasonality and fasting factors, which is in a way expected given that we're dealing with plasma. There's a lot of circulating blood cells that are in there, as well as circulating proteins, which may also be involved as well. But in addition, I think things to look out for are really which sort of cohort you're dealing with. Are you dealing with the randomized selected cohort, or the consortium selected cohort, or the combined cohort? Another sort of ancestry and ethnicity, although that can be proxied with genetic principal components when looking at adjustments for that. And in terms of batches, we haven't seen really massive or really strong batch effects. In general, for the majority of the proteins, the batches tend to be quite clustered together within across the board. So there didn't seem to be any specific clustering of any specific batches there that we observed. And you can see that the UK Biobank, where the blood is sampled from, the center at which people are attended to, we don't see any strong outliers there across the majority of the PC1 and 2 components and others across the sampling centers. So by and large, these batch and center effects seem to be relatively okay across the majority of the proteins. And the other thing to also bear in mind is the time between the blood draw and protein measurement, i.e. the sample age, can also potentially for some of the proteins play a role. And obviously, if you're analyzing the genetic data, the source of the genome typing array, and whether you're dealing with the imputation versus sequencing data is also important. Bearing in mind that exome sequencing imputation does not always give you the same sort of results. So it is always bearing in mind to check some discordances and discrepancies there if present. And obviously accounting for genetic confounders is also quite important. And if you, depending on which disease areas people are diving into and the scientists are diving into for specific traits, there could be other measured confounders specifically related to those diseases, which should also be taken into account. And that is, lastly, obviously, is the protein distributions and transformation. As Cindy noted, is the MPX is a relative measure, so the effect sizes should be interpreted in the context of that, and also is on the log scale. So obviously, if people want to compare like for like with orthogonal assays that are on UK Biobank, do be wary that the log versus linear scale needs some transformation required. And if you we seem to find that inverse rank normalizations seem to be quite pragmatic and does a reasonably good job if people are doing protein-wide scans. But obviously, that doesn't preclude other sort of transformations for specific proteins or for specific hypotheses as well. So a lot of these QCs and also the genetic associations and PTM mappings are mentioned in quite a bit of detail in the supplementary informations for the preprint, and hopefully there'll be even more details in a few months to come in the peer-reviewed journal that's coming out, hopefully as well. And obviously, there's in the proteogenomic field, there's a lot of excitement, with the expanded sample sizes and a lot of emerging themes and patterns that you can do with the data. Some of it sort of are plotted in figures that you might find in the preprint in terms of Mendelian randomization, co-localization, PTL networks, and gene-gene interaction effects that people might be observing. But obviously, proteogenomics is not the only thing that people can do with proteomics, obviously. And there's a lot of non-genetic proteomics-based applications and associations that are possible because of the in-depth phenotyping linkage that you're seeing with UK Biobank. So obviously there's a whole host of things that people can do with these proteomics data, whether looking at biomarkers, associations, or even using proteomics to predict things. And also I think the combining with these other omics data sets, such as metabolomics and emergence of single cell sequencing, could also be quite important as well, in addition to the whole host of imaging derived endpoints and other omic data set that are already within UK Biobank. And obviously it's important to know that this is, despite being a really good discovery data set, it can also be used as a validation for confirmation for people who are using proteomics or Olink-based platforms in other studies as a validation replication data sets. And it's also important to know that these proteomics are done in plasma, so it may not necessarily transcend to other tissues and fluids as well. So, a bit of a, in summary, when working with the proteomics data, please do consider the sources of systematic, technical, but also sample-based and the sample cohort selection that may potentially confound or boost your results as well. But also careful framing of the assay in the right technical and biological context is important. And do beware also the pitfalls of over-adjusting, because there could be potential colliders as well that may crop in and give you false associations through the collider biases if you just over adjust for everything that you could think of. And obviously replication is always the most important aspect. So obviously think UKB-PPP not just only as a discovery platform, but also as a replication resource as well. And obviously that's the open access in the nature of this is really to also facilitates the wider scientific community, I'm sure, with a wide range of applications for this data. And hopefully there'll be many more studies based on this from the wider scientific community coming out in the next years, hopefully, in the years to come. So obviously just to be careful, because obviously with these great data comes a lot of great scientific responsibility and rigor that may be needed. So I'll hand over to Karsten, who will give you a bit more flavor about things you could potentially do with the data that probably are really exciting. So over to you.
Hey, thank you, Ben. That was great. And it's really a pleasure to be here. I'm just very briefly trying to say, well, why am I here talking about this? I think probably because I now represent maybe the user side of UK Biobank and the rep. So we'll come into that. Let me but first start with the more general topic of crossing proteomic data with other data types. So I come from a field where I really try to have my own mini Qatar Biobank, if you want. So we collected about 400 samples here in Qatar with a lot of aliquotes 10 years back. And we tried to do initially metabolomics on them. And this would give you a little bit of an idea of what UK Biobank could become over the time. So it would include all the different metabolomics platforms. We heard already about Nightingale is there, Metabolon, Biocritis, the non-targeted platforms, Genomics. In our Qatar platform, we really have now 20 different platforms, from genotyping over methylation. I heard there's methylation planned for UK Biobank as well, so watch out for that. Transcriptomics data, we have whole genome sequencing, and we have microRNA data. And let me just quickly go through that. So what I show here in the middle is basically how integrating all this multi-omics data really give you a more and more sharp picture of the people you want to look at. So let me get quick over this. So the different platforms, is it coming here? Yeah. So you see, we've tried the Olink platform, of course, and I think people have been talking also about mass spectroscopy data and immunoproteomics. So if you want to get a feeling for what kind of omics data could come to UK Biobank, hopefully in one day, we have this on a web server here, so I don't want to get much deeper into that detail. And now I want to really turn into how can we integrate the UK Biobank data. So what you see here, this is still from my study, where we have the different omics phenotypes. So you can actually integrate the data by doing correlation networks, partial correlation networks, mutual best hits, GWAS hits, EWAS hits, and so on and so forth. But before you can do this, the question is really like, how does the late data look like? And I think this is really where everybody has been waiting for. And the first contact with the UK Biobank data, actually, I had it 2 days ago when the data really got online. And I just want to walk you through that a bit, how this really looks like. So if you're already a researcher, you probably know this website. You log into the web server, you go into the showcase. And what is really new now here is this extra tab with Proteomics Data, right? And once you get there, you would get all the information that's in the data set, and you will see here the data field 3900, and that gives you basically how many proteins are available per sample at the time. And when you know a little bit to read these things, you see the number of participants, the number of items. So some have been measured in multiple instances, and so on and so forth. And then you can click deeper into the thing. And here is the link where you can actually see all the proteins that are in the database, which are a little bit misleading at the moment, because this is the whole data set that will become available in November with the 3,000 proteins. At the moment, it's 1,500. I think a very important resource that initially I always overlooked, so I would like to really draw your attention to that, is there's a lot of documentation already on the website from UK Biobank, and you will find, for instance, the work from Ben Sun, what he just showed, the whole quality control documentation. You can download it as a PDF. And you will also find all the different details like plots on PCA, where the samples come from, and so on. There's also documentation from from Olink themselves. So what Cindy showed a bit earlier, so you would have all the documentation about the plate layout, how the data is structured, and how the QC is done, and so on. So you can download all this. There is also the QC data from Ben. You will see excellent QC in terms of interplate and extraplate variability, and also data points passing QC. And now this, until here, we were still in the framework of the UK Biobank platform. Now you can access the data directly from there, put it into a basket, and have it delivered in the usual form. But what I would really recommend is moving on to the DNAnexus platform. So all the slides here, the links are there. Once you get onto the UKB wrap, or what I call the DNAnexus platform, you can actually go and download, or basically, well, dispense the data. So once you're there, if you have already a project, you have to update it, and it will dispense the Proteomics data to you. And then you can just go into the cohort browser, and I think Ben might say a bit more about that later. For instance, what I just did here, I was just trying to see how can I connect the the Nightingale data and the Olink data. So for instance, you could set filters like, I want to have the number of proteins measured greater than 0, and also I want to have it measured in at least one spectrometer. So that would be two parameters I could set. Both I choose here instance 0, so the take-in study of UK Biobank. And in this case, you can see there would be 1,300 samples that actually overlap between Nightingale and Olink, which is what you would expect, because at the moment there's like 20% of the UK Biobank samples are measured on the NMR platform. And then you can look at the data in detail. So for instance, I choose here to go for the protein ApoM, and you will see the different instances, 0, 1, and 2, and 3, the distribution of the data. And then you can go and download the data, for instance, if you want, using the table exporter tool. And that data, I really, as I said, I just did it 2 days ago. So I really very quickly did a download of ApoM and one lipid parameter from the Nightingale platform. You would get 2 different tab-separated files. You could open them here, for instance, in R, and for good measure, make sure that the identifiers match. And what you see here is then the correlation plot between these 2 parameters. what Ben said before, with great data comes great responsibilities. So you see the p-values here are really crazy. Although when you look at the plot, the correlation is far from being very strong. So there's really a lot of things where you have to get used to the kind of p-values you get when you work with this kind of data. All right, and then just as a very quick teaser, and then I will be offline. So how to work now with the data, how to integrate the different things. I want to make a little bit of advertisement for my colleague Jan Krumzik from Cornell in New York. They developed tools to do, for instance, Gaussian graphical models, to do correlation networks between, they worked on metabolites. I'm really curious to see how that works between proteins. Then they have methods to actually focus onto areas of interest. There's so many phenotype in UK Biobank, so you could imagine you would have your correlation network between all your proteins, between all the Nightingale data, the upcoming single cell sequencing data, the clinical biochemistry data, and then find out what are basically the proteins or the other traits that associate with a given trait of interest, and then highlight these. They have tools also to focus on whether you want to focus on single traits, pathways, networks. And then with this tool, you could then just say, where would I cut my correlation networks or my hierarchical clustering based on the information content with respect to my phenotype of interest. And then in the end, getting away from these hairy balls to really informative network integrations that give you a pathway reflection of what's going on in your data. And as I said, I mean, the data is so fresh and so new. I think with 30,000 users working on the data, I'm really excited to see what comes up. So I will stop here. Just as a really last thing is in terms of software, Jan has developed a toolbox for that called Maplet that allows to actually process the data in such a way that you can just download it in one very convenient form, summarized experiment in R, and then just put different filters in a pipeline form on it and just go and analyze the data in one go. And so this is not from my group. This is a general thing in R. I really recommend once you extract the data from UK Biobank into your own environment, if you're an R programmer, summarized experiment is really a data structure that's of very much help if you want to integrate different data types. And with this, I just acknowledge my founders. And of course, I'm very happy to be part of this. And thank you very much for your attention. And I hand over to Ben.
Awesome. Thanks, Karsten. Really appreciate that. And so really, I wanted to talk a little bit about a call to action, not to just use the proteomics data in and of itself, but to really think about it in terms of aligning it with the other data in the rep. I mean, that to me is what is really exciting. There's 14 years of phenotype data, and there's a wealth of genomic data as well as metabolomic data, which Karsten just mentioned. So what I'd like to do is spend just 2 minutes with a call to action to analyze the proteomic data, alongside that data, whatever disease or technical consideration you're interested in, then give you a couple technical tips and tricks. Some of the other presenters asked me for those along the way, so I'll give you a couple technical tips and tricks, and then we'll get right to the Q&A. We have over 70 questions already asked, and I'd say also check out the related content module. I'll mention that again. But first, really a call to action, thinking about really a number of interesting use cases. First, really thinking about hypothesis confirmation. I think when a lot of us think about biology now, we really think about pathways. And one of the nice things about the Olink platform, and Chris will talk a little bit more later, I think, about why the PPP chose the Olink platform. But one nice thing is that they have representatives in pretty much every major transcriptional module. And that's super cool. And I think that can be done really for hypothesis generation if you believe that a number of transcriptional modules are involved in your analysis. And somebody in the Q&A brought up the single cell data that'll be coming out towards the end of the year, and being able to overlay with that. And I think that'll be even more confirmatory. It'll be very exciting, that first tranche of single cell data. Also, I want to talk about sort of more complex analyses, really doing GWAS with metabolite, and thinking about Mendelian randomization. And I think that the proteomic data will also be amazing for hypothesis confirmation there. So that's really, really exciting, and some things to think about. And again, really a call to action, not just use the proteomic data in and of itself. Of course, interesting things will be found there, but do you really think about using the proteomic data alongside other data? And I think one of the most exciting use cases right now is thinking about disease subtyping. So certainly folks that do subtyping for colorectal cancer, if you think about consensus molecular subtypes, there's already been work done on proteomics, and being able to cross that with drug efficacy, for example, in the UK Biobank, in my opinion, is really, really exciting. As well as looking at things like the onset of blood cancers. So those are all, in my opinion, really exciting examples of ways the proteomic data can be used with other data in the UK Biobank. I have a lot more to say about that, but really I want to move to tips and tricks so we can get to Q&A and really have a discussion of your questions. And we'll probably run for another 10 to 15 minutes after the official end of this webinar. If you can't stay, don't worry, you can pick it up on the recording. So that said, I want to talk about some tips and tricks. Right now, this webinar has been extremely popular. I'm really excited because that shows the enthusiasm people have for proteomic data on the UK Biobank. So that said, pro tip, if really what you can do is you can dispense to a new project instead of refreshing. That's going to be rock solid. It's going to get you the data there in real time right now. So give that a shot. And you can always, you'll still have the genomic data there. And if you have ancillary files, you can copy them from project to project without accruing additional cost. So check that out. That should be big. I think I've made a fairly compelling case for working on the data alongside genomic data. So that's I think, a big deal. Cindy asked me a question about R scripts later. Several folks mentioned scripts and so on and so forth. And really, I'd encourage you guys to go to the tools and tutorials tab and look at the Git book made by my colleague Ted Lederis, where he talks about a lot of things in terms of Bash for bioinformatics. For some of you, it's going to be pretty simplistic, but it gives a great description of wrapping apps in JSON. And I just want to say, and you can also look at question 48 here, Brenton's put some things in the related content module, but really R scripts can be run on TTYD or Cloud Workstation. They can be run in the Jupyter Notebook interface. But ideally, if you're doing large-scale stuff, you can build applets with them. And we have webinars all over the place describing how to do that. Or if your institution would like a sort of quick workshop on how to build apps and applets, or just get leveled up on the UK Biobank, reach out to our team, talk to us, and also reach out to the community. People can point you to the appropriate resources. We can point you to the appropriate resource. So that said, really, I want to plug these two webinars. There were a ton of questions asking about tutorials for the proteomics webinar. Our colleague Alex Lee on June 1st is going to give a webinar on analyzing the UK Biobank proteomics data. I think it's going to be amazing. But to be absolutely clear, the data is available now, both in the UKB rep and in Showcase, and you can grab that right now. But another really exciting thing we'll have is May 25th, we'll have an oncology researcher roundtable with a bunch of people, including my colleague Yin Kao, who is interested in early onset cancers. That's going to be really exciting. as well as some folks from the UK Biobank and elsewhere. So please, please, please check that out. That's going to be really exciting. And so without further ado, what I really want to do is get to the Q&A. And I'd really like to kick off with a question we had, which talking about the whole genome sequencing. And so whole genome sequencing is going to come out towards the end of this year. We'll go from the 150K to the 450K right around the end of November, beginning of December. And so really stay tuned for that. And then as a result, you'll have an overlap between all this proteomic data, the next tranche of proteomic data, as well as the rest of that data. So I'd like to invite Naomi. It'll be 490K, the WGS. Now I'd like to invite Naomi back to the floor and ask her if she wants to comment on this and some of the other questions, specifically about having to get into specific fields, and also whether the single cell data will overlap with proteomics.
Yeah, thanks. So the whole genome sequencing data will be made available we anticipate in November of this year for almost the full cohort, about 490,000. The only reason it's not the full cohort is for a small minority of participants, we were unable to collect a blood sample. So that's the reason it's not the full kit and caboodle for everybody. And the DNA sample that we use for both the genotyping, the whole exome and the whole genome was taken from the baseline blood sample for which we extracted the DNA from. Question around the single cell RNA and the overlap with the proteomics data. The short answer is there is no overlap, because the 5,000 samples that were done as a single cell sequencing pilot, we collected fresh blood samples in order to perform that assay, and that was done fairly recently.
But there would be overlap in a few of the individuals, just not the samples themselves. Is that correct?
That's correct, yes.
Okay, great. So thank you, Naomi. And so then there's another question, really, and I think this is directed towards Chris, but if Naomi wants to weigh in on this, that'd be great too, which is, why was Olink chosen versus another affinity-based technique? And I think Chris is ready and willing to answer that question.
Going to be a long answer, so apologies in advance. So yeah, I think this was 2020 was when I first proposed the idea to conduct this study alongside Melissa Miller of Pfizer and Lyndon Mitnall of Regeneron. And at that time in 2020, there were really only two high throughput proteomics technologies that were capable of scaling to tens of thousands of people, Olink and SomaLogic. And I'm going to point towards the literature a little bit to answer this. There have been several papers now by Mike Pietzner, Claudia Langenberg, Rob Gersten, and others. And those have indicated that there are pros and cons to both platforms. The general consensus is that neither platform is better than the other necessarily. Olink maybe has an edge in terms of protein abundances, whereas SomaLogic might be more sensitive to protein shape or changes in activity. There isn't a huge amount of overlap between the platforms in terms of the unique proteins that they capture. I think it's something less than 2,000 proteins that overlap in the Venn diagram between the two. So the rest is actually unique to each platform. That Venn diagram has a lot of unique Olink proteins and a lot of unique SomaLogic proteins. So overall, Olink and SomaLogic, I think they provide synergistic insights, and in an ideal world, we conduct both, right? But we don't live in an ideal world. We had to choose one. It was difficult. It was difficult. We would choose both if we could. But at the end of the day, if this was purely driven by scientific merit or the assay quality, I don't think we'd ever come to a conclusive decision. But ultimately, I think the overall cost of the study and issues around intellectual property and contracting really influenced our decision to choose Olink. And I think the last thing I'll say is the Olink team was incredibly accommodating and willing to meet the consortium's many complicated demands for this project. So, you know, very, very easy team to work with. So it doesn't preclude us working with other technologies in the future, but hopefully that gives some context for why we chose Olink for this particular project.
Awesome. Okay. So this is a question, I think, for either Chris or Ben, which is, will there be an indication of which subjects are from the randomized baseline versus the consortium selected groups?
Ben can probably weigh in as well, but I mean, there should be, because we received that indication, so the public or the UKB community should as well.
All right. Awesome. Great. Thank you for that. So here's another question for Ben, if he's available, which is, could you please clarify your point regarding random sample enriched rare disease? A random sample would represent the population prevalence and would not necessarily enrich any rare disease. I would appreciate your clarification on the matter.
Yeah, so the randomized component, sorry for the miscommunication there, but the combined component that includes both consortium and randomized would be enriched for diseases. If you just take the randomized component itself, for the vast majority, it's just comparable to UKB, with a couple diseases that are slightly enriched, but not to an extent that there's systematic cross the board. So yes, you are correct that most diseases are not enriched in a randomized baseline, as you'd expect.
Cool. While we have you on the line, there's another question, and there's been a lot of question about batches. And I think this is one of the well-worded ones. Could you elaborate a bit on how the PPP deals with bridging sample selection between batches and between kits and lots?
So yeah, it's a good question. So a lot of it is in the detail in the supplementary information. But as a brief overview, there are bridging samples placed across different plates in the wells. But the normalization procedures initially was based on the mean normalization across the different plates, because most of the samples are randomly scattered across the different plates. So they assume that the mean are comparable, and the bridging samples are there used to check the calibrations of the normalization to see if they're within tolerable ranges.
Cool. Thank you. There's a couple quick things I might be able to help people with, so I can answer them myself. One is, why would you want to dispense a new project instead of refreshing? I would say that right now, the refresh is rather slow because of the amount of people refreshing their data, whereas dispensing is still quite fast. So for refreshing, you would probably have to wait a little bit, but if you redispense, you can get the data pretty much immediately in 4 to 24 hours. Yeah, that was asked twice, so I think I just covered that. And then there was, so maybe this is a good one to take us out, because I like this question quite a bit. And this is again for Chris and Ben. Are there any plans to make a GUI browser? I guess maybe this is also for our team, but are there any plans to make a GUI browser for the BQTL data?
It's actually a question for Karsten, because Karsten's been helping us build that with Matthias and Gabby and others. So maybe Karsten.
Cool. Karsten, do you want to weigh in on that?
Yeah, well, I didn't want to preclude on that. Yeah, actually, it's the team of Gabi Kastenmüller who already created many very great browsers for the previous study on SOMA and on Olink. And her team is presently working on that one, and that one will also become online when the paper gets published in a journal.
Fantastic. All right. Well, I'd like to say really thank you to all the presenters. Thank you to everybody for making this a really active webinar. We've had something like 86 questions. If we didn't get to your question, we'll try to get back to that. As always, feel free to ask questions on the community.dnanexus.com. If you have a technical issue, you can always write to support@dnanexus.com. And really, I think it is amazing that everyone here is really sort of pushing the envelope of what we can do with bioinformatics. So, all right, well, with that, thank you everybody for being here, and have a great rest of the day and a great rest of the week.